All right, hi everyone, and welcome back to another CBRG session. This session will be the first of a series dedicated to single-cell RNA-seq analysis. And we will begin today's session by just going over some basics of single-cell RNA-seq, and then we'll dedicate most of the time to coding. So the book that we'll be using as our guide for this kind of mini-series is this book here, the Single Cell Course book produced by the Wellcome Sanger Institute. It's a great book. It provides a comprehensive introduction to single-cell RNA-seq analysis, and it's associated with, I believe, a 16-hour long course that was held, I think, in Cambridge a couple of years ago. So we won't be spending 16 hours on this because we'll be skipping most of the introductory chapters that have to do with a very comprehensive introduction to the technique of single-cell RNA-seq analysis and things like data preprocessing, genome alignment, quality control, things like that. So to begin, so we can see this figure here in chapter 2. So this shows you a kind of high-level overview of single-cell RNA-seq. It compares what's known as bulk RNA-seq or just RNA-seq up here with single-cell RNA-seq. And we dedicated a few sessions a couple of months ago to RNA-seq analysis. So the good thing is a lot of what we did learned there can be applied to single-cell RNA-seq analysis. So as you can see here in bulk RNA sequencing, if you have a bunch of cells, so if you have heterogeneous tissue, what you do is you just pool all of that RNA and you get kind of an aggregate overview of view of gene expression. But with single-cell RNA sequencing, as you can see here, the transcripts are of different colors, indicating that they come from different cells. So you can get expression quantification at the single-cell resolution. So this is another nice figure, also from chapter 2, that goes through the entire RNA-seq, so single-cell RNA-seq process. So you begin with heterogeneous tissue with different cell types that undergo some type of disaggregation, which could be enzymatic or mechanical, and then are separated into single cells. Now, this could be done by, for example, flow cytometry. And these cells are then captured, and we'll go over a few of the popular approaches that are used to capture these single cells. They're lysed, and then we collect the RNA. The RNA is reverse transcribed into cDNA, and you see here, it's slightly difficult to see, we add what are known as UMIs, which are unique molecular identifiers, and cell barcodes to these sequences. And the cell barcode, as the name suggests, allows us to keep track of the different reads. We can tell, based on the cell barcode, which cells the read comes from. And the UMI, that's a little bit more difficult to explain without a diagram, so I will explain that in a couple of minutes. So we prepare our library, and so that we can see here in panel 3. So we just have a list of sequences with the barcodes, UMIs, and the actual RNA. And then there's a process called demultiplexing, which is a way of, as you'll see with these UMIs, what you do is you collapse. You do this thing called deduplication, which I'll describe in a minute. And you can assign the different transcripts, the different reads to different cells. So that's this process here. And you then just count the number of UMIs, and you align them to a reference genome, and then you do that. By doing that, you produce this matrix of counts. And this is similar to the RNA-seq count matrix that we saw, where in this case, the rows, again, are, for example, genes, but the columns, instead of being samples or individuals, are individual cells. And then finally, we get to the fun stuff of actual data analysis, where you can do lots of things like dimensionality reduction by PCA or TSNE or UMAP, and clustering, differential expression analysis, trajectory analysis. We won't be covering all of this, but we will touch on dimensionality reduction, clustering, and differential expression analysis. So I mentioned that there were a few ways to isolate these single cells. One is this microtiter plate approach, where you use something like flow cytometry to separate individual cells into individual wells of a multi-well plate. The second approach is this thing called microfluidic arrays. So these are chips that that you can use to basically, as they say here, trap individual cells in, I guess, individual wells. And then you can do the process of reverse transcription, adding a UMI, adding a cell barcode. And the data set that we'll be using today, and actually for the series, was generated using one of these microfluidic arrays, so using the Fluidigm C1 platform. And what is now the most popular approach is this microfluidic droplets approach. So this involves using these beads that have distinct barcodes and associating individual cells with separate beads. And this approach is the 10x genomics, the chromium approach. And it's the most high throughput and right now the most popular approach to doing single cell RNA sequencing. I mentioned this concept of UMIs. So this is a nice figure that shows you different sequences. So that's these dots here. The barcodes that are associated with these sequences, and as you can see, in this case there are 4 cells out of thousands. You can see that for each cell has the same cellular barcode. So that's how you can tell which transcript came from which cell. Along with that, you have these things called UMIs. And as the U in UMI stands for unique. So these are unique to the different transcripts. And where they are especially useful is, so I should back up a bit. The problem with single-cell RNA sequencing is that you start off with very little RNA. So you have to, once you do reverse transcription, you have to subject the cDNA to multiple rounds of PCR. The problem with PCR is you can have amplification bias. So some transcripts amplify or replicate more efficiently than other transcripts. So using these UMIs is a way to kind of account for this bias. So once you produce all of the reads by this PCR, what you do is you find all the transcripts. So for example, beta-actin here, ACTB, all the reads that map to that gene, you find all the ones that have the same UMI. So this implies that all of these transcripts came from the same original molecule. And so what's done is those transcripts are then, those reads are then collapsed. And so in this case, there are 2 reads that have the same UMI, so they get collapsed into one count. Conversely, you can have another gene, so you have ARL1, so you have 2 reads that map to that gene, but they have separate UMIs, meaning that they originated from 2 separate molecules. So in this case, you have 2 reads, but these 2 reads correspond to 2 molecules, so you have 2 counts here. So the point of all of this is the data set that we'll be using is a count matrix, but the counts aren't of reads, they're of these UMIs. So that's something to just bear in mind. I mentioned this data set that we'll be using. It's produced by Tang et al., and it was specifically designed, this experiment was specifically done to characterize things like batch effects, sources of experimental noise, and other such unwanted sources of variation. And so here they processed 3 C1 replicates. So they used 3 human iPSC lines, and each iPSC line there were 3 technical replicates. And so this way they could compare technical replicates with biological samples. And they use this Fluidigm C1 platform. And before we move on to so that's a 9-minute introduction to single-cell RNA-seq. I would strongly recommend that if you aren't particularly familiar with single-cell RNA-seq or you want a refresher, chapters 2 to, well, really chapter 2 is what you need. Chapter 3 is great if you want to know more about all the steps that are between collecting the RNA and getting this count matrix. So we won't be covering this. And this is just like with the RNA-seq series we didn't cover pre-processing. So once we produce this, I mentioned we get this count matrix of, in this case, genes and cells. So that can be seen here in the middle. This is this assay. Now, this slightly confusing looking figure corresponds to the structure of the single cell experiment object. And I mentioned that a lot of what we will be doing in this series is very similar to what we did with RNA-seq. And that's largely because the single cell experiment object structurally is very similar to the summarized experiment object, which in turn is similar to the expression set object that we took a look at with microarrays and the Q features object that we took a look at when we did our proteomics analysis. So hopefully you can now see, now that we've moved on to our fourth omics analysis, this kind of unifying effect of using these different objects that are all structurally very similar. So just to remind you, just as a summarized experiment object can hold one or more assays, so can a single cell experiment object. So we will begin today looking at we'll begin with one assay, and that's just our raw count matrix, so our UMIs. And then we can do all kinds of things to it. We could log transform it, normalize it, do whatever we want to those data. And we can use these transformations to produce multiple assays. So in this case, that's what this is meant to show, that you can have, in this case, 2 assays. You can have many more than 2 assays. And then using this row data function, you can look at if there are any data associated with the genes. So for example, maybe your data set, in addition to having the gene symbols, also has entree IDs, ensemble IDs, genomic locations, things like that. And similarly, you can see if your single cell experiment object has any column data. And so since the columns correspond to cells, this corresponds to cell data. So you can learn, so these are just individual cells. But I mentioned that in the Tung et al. Data set, all the cells come from 3 individuals. If you want to know which cell came from which individual, you can use this call data function. And then finally, when we move on to dimensionality reduction, by doing PCA, for example, we can get the reduced data that can also be included in the single cell experiment object. So this is a versatile object that can hold all of the necessary information associated with our single cell experiment, and it is a wonderful way of keeping track of all of these different but related pieces of data, which you would struggle to do if you used Excel or something like that. So that is a quick fire introduction. I'm going to move on to the coding component. So I'll move to RStudio. And as usual, let us navigate either to the project folder the project directory associated with these meetups if you have one, and if you don't, just open up RStudio. And once you've done that, you just go into the console, and as usual, we type in remotes, install _github. And then in quotation marks, do duke cct /cbrg. And cbrg is in all caps. I'm going to just increase the size of, okay, hopefully everyone can see things more clearly now. And then we set the force parameter to be true. And this, of course, is in case you have attended previous sessions. What this does is it will override the project, package that you already have installed. Now, what I have done, which isn't best practice, is I have included the necessary data files that we will need for this session and the rest of the series in the package. And so that's made this package kind of heavy. So, and because of that, it's taking a little while to download. But hopefully, it should download without any error, as it has here. So once it's downloaded, you just load it the usual way, as you would with any package, library cbrg. And then you use the launch project function to launch today's RMD, which is number 13, because this is the 13th meetup. So if all goes well, you should see an RMD file with this title, 13_project. If you scroll down to you see this huge setup chunk, and all of this code, as usual, was written by Paul. And you can see that there are many packages that we'll be using, but this is not, thankfully, not just for today. We'll be using this for the remainder of the series, and these are all, many of these packages are useful to have installed. So we'll go through the trouble of installing and loading them. So just run this chunk. In my case, I already have all of them installed, so what this chunk is doing is just loading them. As you will see, so please keep an eye on your console, this will likely take minutes to download and load. And of course, as usual, R will ask you for a bunch of things in the console. Just say yes to just agree with everything. So I'm going to go ahead and move on with the rest of the code. And even though you might still have packages being downloaded, because this is being recorded, so in case I go a little too fast or I'm ahead of you, you can always go back to the recording. So I've put in here what I think are some useful links. So the first one is to the book itself. And then I've shown you where I've downloaded the files that we'll be using today. And then I gave a super hand-wavy, quick description of what UMIs and barcodes are. If you want to sit down, if you aren't familiar with this, you'll probably need to sit down and go through the details. And this link here is to a training course that was done at Harvard that explains all of these things in a really nice way. And finally, here's the paper, the link to the paper whose data set we'll be using, the Tung et al. Paper. So let us begin by creating a single cell experiment object. So to do that, we'll first need to download two data sets. One is the data set that contains our count information. So this is the count matrix. And we'll also download a data set that contains information on the different columns. So remember, in this case, columns correspond to cells. So let us go ahead and do that. So we go to line 99 and just run that. So if you want to run a single line, you just hit Command Enter or Control Enter, depending on whether you have a Mac or a Windows machine. So we have, if we look at our environment pane, we see that we have a data frame called tongue_counts. And similarly, we have tongue_annotation. We can just take a quick look at them by clicking on them. So we see tongue annotation has all of the individuals in the first column, and then it has additional potentially useful information like replicates, wells. So this is corresponds to the individual wells in the Fluidigm C1 chips. And then we have batch identities and sample identities. And if we look at the tongue counts, this is a much bigger data frame, so it'll take a little longer to load. We see as row names, we have these gene IDs, and the columns, we have the cells. And you can see that typically for single-cell RNA-seq, these are giant matrices that are referred to as sparse matrices, because as you can see, most of the entries are 0, meaning that there are, so for example, this gene is not detected in this cell. And yeah, that's it. So this is the count matrix that we'll be using. So taking those two, so actually let's quickly inspect the data. So one quick way to do it is to just use the dim function. So if we do dim underscore counts, and we run this, if we look at the console, we see that there are 19,027 rows, so meaning 19,027 genes, and there are 864 columns, or 864 cells. Similarly, if we do dim tongue underscore annotation, oh no, sorry. Let me do that again. We see that this has 864 rows and 5 columns. So if you look at the two, if you compare the two, you'll notice that the number of columns for tongue counts is equal to the number of rows of tongue annotation. And this is essential because the columns in this data frame correspond or must correspond to the rows in the second data frame. Those two must match. So that's the answer to this question here. What is the relationship between the two objects above? So now that we have these two pieces of information, creating a single cell experiment object is super easy. Use this function. So we go to line 109 called SingleCellExperiment. And we can see that it has two parameters. One is called assays, which, as the name suggests, stores our assay data, which in this case we just have one assay. So these are the tongue counts. And then we have our column data, call data. And that's the tongue annotation. So call data is easy enough. You just do call data is equal to tongue annotation. For the assays, you have to do a little bit more work. You have to first, let's look at this. Let's go inside out. So we begin with tongue underscore counts. Now, as I mentioned, if you hover over tongue underscore counts, you see that it is a data frame object. And single cell experiment does not take a data frame, it takes a matrix. So to convert this data frame to a matrix, you just wrap that in an as.matrix function. And then you have to give that matrix a name. So we can call it counts. Counts equals as.matrix tongue underscore counts. And then in turn, we have to put all of this inside a list. And the reason we do that is because I mentioned that this single cell experiment object can hold more than one assay. So if you happen to have already, for example, log transform data, and you wanted both the raw count data and the log transform data, you could just put those two pieces of information within this list. Okay, so if we run all of this, and we can assign all of this to this object called tongue, T-U-N-G. Now, so this line here, before we take a look at the tongue object, if we look at line 115, in the book, they say that we now no longer need our original two data frames, tongue counts and tongue annotation. So there are a few ways to get rid of it. You don't have to get rid of it. I kind of like in the book that they recommend getting rid of these two because it just makes our environment pane less cluttered. And as you know, whenever you're doing any kind of analysis, this environment pane can get crowded really quickly. So to remove it using code, you just use this function called rm, which stands for remove. And you do rm and then give it the objects you want to remove. So if we run that, we now see that those two data frames have disappeared. And we're left with just this tongue single cell experiment object. So let's take a look at it, line 117. So if we run that and take a look at our console, if you attended any of the previous sessions, this kind of format should look familiar to you. This is exactly what the summarized experiment, not exactly, but this is very similar to what the summarized experiment output looks like. You see that it shows you the class of the object, the dimensions, number of rows, number of columns. Is there any metadata? No. How many assays does this have? One. And then it gives you some other potentially useful information. But obviously, this is just a snapshot of this giant object. So to take a look at individual elements of this object, you will have to use these so-called accessor functions. And these functions are functions we've come across previously. In fact, they're the same. Many of these are the same functions we used for the summarized experiment object. So I mentioned that along with the assay data, we can have we can have row data. So that's data, I hesitate to use this word, but let's use it. It's metadata associated with the genes, and column data are metadata associated with the cells. So if we do use the function row data, capital D, tongue, So line 190, if we run this and take a look at our console, we see that we get an empty data frame because in this case, we don't have any row data. However, if we go to line 121 and run call data, we see we do have some data. So this should look almost identical to the tongue annotation object that we've now deleted. And so it shows you the individuals, replicates, wells, so on. And this call data will serve as a location for other useful summary statistics that we'll be calculating today. Now, if you want to take a look at the raw counts, you can use this function called assay. So if you go to line 122, an assay takes in the name of the single cell experiment object, so it's called tongue, and then, comma, the name of the assay. Now, in this case, there's just one assay in this single cell experiment object, and it's called counts. So if we run all of this, we see it doesn't output very well because it's a huge matrix that we're outputting of gene and cell. It's a gene by cell matrix with all of these counts. And if you want to access an individual column from the column data, that's easy enough to do. So if we go back to call data and we run that, we see that one of the columns is called individual. What if you just wanted to get a list, or rather a vector of these individual entries? That's easy. If you go to line 123, you do tongue $individual. If we run that, we see, so these are the names of all the individuals in the data set. Now you can see that there are lots of repetitions because there were only 3 true individuals to start with. The reason there are all of these repetitions is because there are multiple cells that were taken from each individual. And finally, you can subset a single cell experiment object just like you subset a data frame. So we go to line 124. Let's say we just wanted information for the first gene, so that means the first row, and the first 2 cells. The second cell. So that'd be the second column. We can just do that here. Now, if you look at the output, it's not very exciting. It doesn't actually show you the, for example, the count data. You'd have to do, maybe save. You could save this as another object and then use the assay function on that object. But this just shows you that when you subset, you now have a single cell experiment that just has one entry because you've just selected the first gene of the second column. So this is not particularly useful, but as you will see, subsetting using more kind of useful conditions is a useful thing to do. Okay, so in the corresponding chapter to all of this, so that's chapter 6, they have some really useful exercises, and thankfully they also have the solutions to those exercises. So let's go through those exercises. So the first question in that chapter is how many batches and cells per batch are there? Does that number make sense? So let's run some code to see how to get the answer and then work through the code. So it's asking you about batches and cells per batch. Now, if you go back to call data tongue, you see that the batch information is in your column data, your call data. So that's a clue. So we begin by accessing this again. So we can do call data tongue, and we can pipe that into. So I'll be using the solution I will show you. Will use a deployer function. So to do that, it's always safe to convert whatever you're using into a tibble first, because you know that once you convert it to a tibble, you'll have no problem using the deployer functions. So we convert it to a tibble, and then we do deployer : I'm going to use the count function. And what am I counting? I'm counting the batch. Now, I could have just written this count. Now, I don't know what packages cause these conflicts, but count, as you can imagine, is it's a common word. So unfortunately, this name refers to functions from packages besides deployer. So sometimes you can get some funny outputs if you don't put in dplyr. Actually, let's just try it to show you what I mean. If I run this as is, I get this. Error in count, argument x is not a vector, list. I don't know what this means. Because clearly this error is associated with some other count function. I want to use the function count from the dplyr package, so I just do this. And now when I run this, I see I get this nice table. So this gives me both answers. How many batches? Well, I can see that there are 9 rows here, so there are 9 batches. And then within those batches, how many cells are there? I can see that the n is 96 for each of the batches. And it asks, does that number make sense? It does because of the platform we're using, this Fluidigm platform. So each batch is associated with a different Fluidigm chip. And each of these chips has a capacity of 96 cells. So that makes sense. 96 cells, 9 chips. And we can confirm, and so that's where, for example, if you go back to this dim tongue, I'm just going to run that in the console. You see the second number here, 864, that refers to the total number of cells. 864 is just 9 times 96. And we can confirm that here. Is this true? Yes, it is. So that's my solution. The book solution is more succinct. The book uses this function called table, which I don't believe we've come across so far in this series. So you just do table, and then I showed you previously that you can access a specific column within your call data like this using the dollar symbol. So you do tongue $sign batch. And if you run this, you get the same output, obviously the same answer, but the format is a little bit different. So here you can see that there are 9 columns, and then each entry, it's actually, sorry, not columns. You have 9 names, and then each corresponding to the 9 batches, and you can see each batch has 96 cells within it. And if you want to learn more about this table function, of course, there's the question mark table. Okay, so let's now move on to modifying the single cell experiment object. So this is where things get more interesting. I mentioned that we can, and just as you saw with the summarized experiment object, when we were doing RNA-seq analysis, and actually, I forget now. Yes, RNA-seq analysis, but also for proteomics analysis, we were doing lots of transformations. And what we were doing is whenever we were transforming a particular assay, we were saving the output as another assay. So we were accumulating assays as we were going through our analysis. So we're going to do something very similar here. So what we will do now is let's log transform our count data. And we'll go into why we want to do log transformation in the next session when we cover things like normalization and transformation. So to perform a log, so when I say log transformation, it's log base 2. To do a log 2 transformation of our counts data, it's easy enough. You can do, oh, I should mention one thing before this. I think I mentioned this later on. So I said one way to get the counts data is to do assay, the name of the single cell experiment object, and then the name of the assay. So you could do this, right? A more convenient way to do it is to use this accessor function called counts. So all you have to do here is do counts tongue, and it gives you the same output. So that's a handy thing to have. And as you will see, that there are similar such accessor functions for log counts and other information, other assay types. So we do counts tongue to access the counts data. So we go to line 151. And before we log transform these data, we have to remember to add a 1. And that's because log 2 of 0 is meaningless. So if we do this, the way to save this information as a new assay is to assign it to, first you do this assay function, give it the name of the single cell experiment object that you want to assign this new assay to, these new data to, and then give the name of the new assay. So since I'm doing log 2 on the counts, I'll call that assay log counts. So if I run line 151, it's going to take a second to run. And now if I take a look at tongue, look at the console, you'll see now instead of one assay, I have two assays. One is called counts, the other is called log counts. So that is pretty simple. And similarly, if you wanted to take a look at just the first 10 rows, meaning the first 10 genes, and the first 4 columns of the log counts assay, you can do assay, tongue, comma, log counts, and then you can use the usual square bracket subsetting. So you do 1 to 10 for the rows, comma, and this is a typo, it's not 1 to 5, it is 1 to 4 for the first 4 columns. And if you run this, you can see you have the log transform data, count data for the first 10 genes and the first 1, 2, 3, 4 cells. I mentioned this helper function business. So once again, just to re-emphasize this, if you wanted to access the count data, you can do it like this using the assay function, assay tongue counts, and you get an output. Or more conveniently, you can just do count assay. Oh, I'm sorry, counts, because what? Oh, sorry. You don't say assay, you give it the name of the assay. Sorry, name of the single cell experiment object. So you do counts tongue. Similarly, if you wanted to access the log counts, you can either do it the long way, assay tongue log counts, or you can do log count. And then just give it the name of the single cell experiment object. Then it'll give you the identical output. It's just easier to write line 164 than it is to write line 163. Okay, so now let's work out some useful summary statistics. And today we'll really just be doing this to show you how to do it. In the next session, we'll show you why we're doing this. So the first thing, line here on 172, it's calculate the mean counts per cell, so meaning per column. So what happens if we just use, so we've used the base hour mean function, right? So what happens if we just do mean counts tongue? We just get one value. Because what mean does is it takes all the values, so all 19,000 times 864 values, and just works out one global mean. We don't want to do that. We want to calculate one mean for each column. We could write a little function to do this. We could use functions that are dedicated to this, like the apply function or the map function from the per package, or we can use a simple function called call means. So we just do call means on counts tongue. We now get for each column, meaning for each cell, we get the mean counts across all the genes. And just to confirm that this corresponds to the cells, we can just work out the length of this vector, this resulting vector. And we see that it's 864, which is the number of cells that are in the single cell experiment object. So that is calculating call means. Now, this is potentially useful information that you might want to keep track of. How do we store this information? Or which part of the single cell experiment object can we add this information to? So one kind of obvious place to add this to is the call data, the column data. And so to do that, we can do call data on tongue. And on the fly, we can create a new column using this dollar symbol, and we'll call it mean counts. Seems like a reasonable name. And to this, we can assign this call means. Right? And now if we run this, now if we do call data on tongue, we can confirm that we now have an additional column called mean counts that contains the mean count data for each cell. Cool. So that is how to calculate some simple summary statistics and how to save those summary statistics in your single cell experiment object. So moving on to the exercises section of this exercise part of this section, it says to add a new column to call data called total counts with the sum of counts in each cell. Okay, this is easy enough. What I'm going to do, because I'm being lazy, is I'm just going to copy paste line 181 here and change a couple of things. I'm going to change mean counts to total counts because that's what it's asking us to do. And instead of using the call means function, I'm going to use the call sums function. And if you want to know what such functions are available to you, if you go to chapter 6, there's a nice handy list of all of these functions and what they do. So let me confirm that I have actually created a new column called total counts. I'm just going to run call data tongue in the console, and I now see that there's a new column called total counts. Cool. So this is where things get more useful. We sort of see the point of doing this. If we go to line 191, it's asking us to create a new assay. So a third assay called CPM. And CPM stands for counts per million. And this is a useful way of representing RNA-seq information, data. So this CPM assay should contain the result of dividing the counts matrix by the total counts in millions. That's a little bit tricky. So I'm going to just show you the book's answer. So actually, let's work this out from kind of inside out. So what is it asking us to do? It's firstly asking us to divide the counts matrix. Okay, so that's just simply counts tongue. It's asking us to divide that by the total counts. Okay, total counts is easy. We can just do tongue $ total counts. Cool. And then it's asking us to do that in millions. So to do that in millions, you just would divide this by a million. So you do 1e6, or you could just write out 1 million. 1e6 is a better way of doing it. Now, so that's the, well, this is the book's answer. This is the CPM data that it's asking you to calculate. But it's also asking you to create a new assay with this in it. So to do that, you can do assay. So just as we created our log transform data, you can do the same thing for the CPM. You can assign that to assay tongue, and then you give the assay a name. CPM seems appropriate. I'm not going to run this because I actually don't think this is the right answer. Because what is it asking us to do? It's asking us to take all of these total counts. So if you recall, total counts has 864 values because it has one count for each sample. And it's asking us to go row by row and divide our count data by the total counts per cell. This line of code doesn't do that. So this is not doing it row by row. And to kind of show you why that is, I'm going to take a slight detour. I hope this is useful. I'm going to go through, one thing is, if you've noticed, if you go to line 194, when you do this, look at the book's answer, what it's asking you to do, it's asking you to take a matrix and divide that by a vector. That seems a little silly because a matrix is a two-dimensional object. How do you divide a two-dimensional object by a vector, which is one-dimensional? Let's take a look. Let's first take a look at how to simply divide one vector by another. So let's take a simple vector, C1, 1, 2, 3, and divide it by another vector. Let's do 2, 4, 6. I'm actually not paying attention to if there are any comments, but I'm going to pause for a couple of seconds and ask you to work out what you think the answer to this is. Ideally, just in your head, or if you want to run it in R, that's also acceptable. So let's divide this one vector by another vector and look at the output. We see that the output is 0.5, 0.5, 0.5. And that's because what R is doing is it's taking the first element of the first vector, dividing it by the first element of the second vector. Then it's taking the second element of the first vector, dividing it by the second element of the second vector, and so on. Here's an interesting thing that R does. Let's now repeat this. So if you do that, run line 196, you get 0.5, 0.5, 0.5, which makes sense. Let's instead make the first vector a little bit longer. Let's repeat elements. Let's do 1 and then 2 again. So now we have a vector that is of length 5 divided by a vector that is of length 3. So I'm again going to pause for a couple of seconds. Do you think this will work? Do you think R will give us an error? And if not, what do you think the answer will be? So 1 second, 2 second. Just going to run this. Oh, that is interesting. Huh. I apologize. I thought this would work. Okay, so it's not allowing us to, so it is an error, that's the answer. It's not allowing us to divide, because this has 5 elements and this has 3, and 5 is not divisible by 3. Okay, so let's make this 6. So what we see here is the answer, again, it's now instead of 3 outputs, you get 6 outputs, 0.5, 0.5, and so on. Because what R is doing is it's taking the elements of your second vector, right? And it's doing the same element-wise division as you saw on line 196. But once you reach the end of this vector, what R does is it just recycles the elements. So it kind of repeats the division. So I would encourage you to play around with this just to see how this works. Let's quickly take a look at matrix division. I promise that this is not some arcane thing. This is actually really useful to understand. I don't believe we've spent much time constructing matrices, but let's make a two-dimensional matrix. I'm not going to go through the details of how I'm making this matrix, but if you want to learn more, you can just do question mark matrix. So, So let's make a matrix that looks like this. So you can see that this has 2 rows and 3 columns, and the values 1, 2, 3, 4, 5, 6. Great. And let's call this matrix mat. And let's create another vector. Again, let's just take this one here. Vec. Oops. Right. Let's now see what happens when we divide the matrix by the vector. What we see here is what R does. So R does things by default. By default, R does things column-wise. So here, what it's doing is it's taking the first element of the vector, dividing it by the first element of the matrix. So the 1 divided by 1 is 1. Then it's taking the second element of the vector and dividing it by this thing here. So this corresponds to row 2, column 1. And then it's going up, doing the division, then down, doing the division, then up, then so it's going column-wise. And again, you can see it's recycling values. So 1, 2, 3, and then again 1, 2, 3. This is what is happening, I believe, this is what is happening here on line 194. And I don't think that's what we want to do. What we want to do is we want to take our vector 1, 2, 3 and divide, firstly, the first row of the matrix by that, and then go to the second row and divide that by the same vector, and so on. That's exactly what I think we want to do here. So how do we do that in R? Here's one really weird way. We can transpose this matrix, divide that, and we can transpose the output of that. We get what we want. Because you can see now we have this matrix 1, 2, 3, 4, 5, 6. If we divide the first row, take the first row, 1, 2, 3, divided by the vector 1, 2, 3, you will get the output 1, 1, 1. Similarly, if you take the second row, 4, 5, 6, divided by the vector 1, 2, 3, you will get 4, which is 4 divided by 1, 2.5, which is 5 divided by 2, and 2, which is 6 divided by 3. So I apologize for this detour, but getting a good sense of how matrix vector, in this case division, works will help to answer this question. So I don't think that's the right answer. So I'm going to give you my answer. So the assignment operation is still the same. You could do assay. Oops. I don't know what to put the answer now. Okay, there. You do this here. And then we can either use that transposing business I showed you, but it's not very intuitive. And especially when you have a giant matrix, it's really hard to picture what the transpose of that matrix looks like. So thankfully, there's a function called sweep that allows you to do this instead of column by column division, row by row division. So the way this works is you do sweep, and the thing that you want, the name of the matrix, so that's counts underscore tongue, and then this number here, 2. So that specifies that you want to do it by row. If you wanted to do it by column, you would do 1. So you do 2, and you give it the name of the vector that you're using to divide. So that's this total counts information. And then you need to specify how you're doing like what function are you doing? Are you doing addition, multiplication? We're doing division, so we just do /. And then remember to divide it by 1,000,000 because it counts per million. So now if we do this, we have a new assay. And how do you access this assay? There are 2 ways again. You can either use the assay function tongue like this. It's going to take a second to run, maybe more than a second. And you can see now that these are all the counts per million values. Conversely, you can use the function called CPM. You just give it the name of the single cell object, and you run it again. Takes another few seconds to run, and gives you the same output. And finally, if you wanted to see, if you type in tongue in your console, you'll see that now you have 3 assays: counts, log counts, and CPM. Okay, we're running out of time. We have 9 minutes left. Let's move on to subsetting. So I mentioned earlier that you can, just like you can subset a data frame, you can subset a single cell experiment object. So you can do it numerically like this. So I'm just going to run through this, and I have the text here that shows you what each of these subsetting operations does. So you can either do it numerically, or you can do it using these character vectors. So you can give it names of genes like this, or names of samples, or names of genes and samples. All right. This is good to know, but it's not especially interesting. What is interesting is subsetting on what's known as conditional subsetting, so subsetting on Booleans. And this is useful because you can subset on conditions, meaning that if a certain cell doesn't match a certain condition, you can use this conditional subsetting to get rid of it. And we'll go into why you might want to do that. So to do that, let's first work out, so in this case, they want us to work out the mean of the counts for each gene. So previously, we worked out the mean of the counts for each cell. Now we want to do genes. So since we used call means to work out the means for the cells, we're going to use row means to work out the means of the genes, because the genes are our rows, right? So if we do that, and we run gene means, let's do gene means, and then let's take a look at its length. This should be identical to the number of genes in our data set, which it is. And then you can take, since this is just a vector, you can take a look at the first few elements just by subsetting like this. And then again, I don't know why I have this here, length just gives you the number of genes. So to show you, let's get rid of this. So we go to line 246. It says we can use such a logical vector inside the square brackets to filter out data, which will return only the cases where the value is true. So this is missing one thing. What it's missing is this. If we use this gene names, which is a vector, to convert it to a logical vector, we can just specify some condition. So I want to specify that gene name should be greater than 0.01, meaning if the mean count data for the gene is greater than 0.01, this will output a true. And if it's equal to or less than this, it'll output a false. So if we run that, gene name's not found. Oh, I'm sorry. I meant gene means. So we run this, we see we get a bunch of trues and falses. So what we can then use, we can use this logical vector to subset our tongue single cell experiment object. And to do that, since these all correspond to rows, we can take this condition and put it here at the start of the square brackets, comma, and we want to keep all the cells, so all the columns, so we put nothing there. And if we run this, we now see, if you look carefully, look at the dimensions, we've gone from 19,000 and something to 16,052. And that's because all the genes that had mean count data that was less than or equal to 0.01 were discarded. However, if you look at the number of columns, so the number of cells, that number stays the same. So it says here, another common use case is to retain cells with a certain number of genes above a certain threshold of expression. For this question, we need to break the problem into parts. First, let's check in our counts matrix which genes are expressed above a certain threshold. So to do that, it's easy enough. So to get the counts matrix, you do counts, and then the name of the single-cell experiment object. And you want to see which ones have a non-zero value. So you just do counts data greater than 0. If we run this, we get, now the output is two-dimensional. And you can see for each entry of a gene and each pair of a gene and a cell, you have either a true or a false. Meaning, is it 0, so false, or is it more than 0, true. And we can see that our matrix is now composed of only true-false values. Because true-false, and then it says, because true-falses are encoded as ones and zeros. So this is something I think we've mentioned before, and if we haven't, it's good to know. You can coerce a true to be 1, and you can coerce a false to be equal to 0. And we'll see why this is useful. Because true-false are encoded as 1, 0, we can use call sums to calculate the total number of genes above this threshold per cell. Let's take a look at that. So let's do this first operation. Let's take, again, working inside out, let's take this condition, counts tongue greater than 0, and then sum it up. So if we do this, let's just run this in the console to see what we get. So these are call sums, so these will be the sums for, for each cell. So you can see here, you have all the cell names, and then you have numbers here. So these are the number of genes that have non-zero counts. So for example, this final sample here has 8,677 genes that have non-zero counts. And the remaining 11,000-ish genes all have zeros. So we can save this information as total detected per cell, right? And that's just a vector. So we can take a look at the first 10 entries. Those are the first 10 entries. And what we can do is we can take this vector and create this condition here on line 266. So actually, let's just take this out. So this is asking which of these numbers is greater than 5,000. If it is, I want you to output a true. And if it's not, I want you to output a false. So if we run this, we get, again, a bunch of trues and falses. So we can use these trues and falses in our column part of our square brackets. And since we want to keep all the rows, we just put a blank before the comma. And if we run this, we now see, if we look at the dim part, we see that we retain the same number of genes because we haven't done anything to the genes, but we filtered out a few cells. And the cells that we filtered out are cells that have a total count, total number of total number of genes with non-zero counts less than 5,000. So we've gone from 864 to 849. So I am going to, since there are just 2 minutes, there's this exercise here. And I'm being a little bit unfair because this is quite a challenging exercise, but the solution is in the book. So I'm going to leave this exercise for you to do, and I will try to remember to go through the solution of this exercise in the next session. The logic we're using is the same as up here, except here, let's just go through the question without actually going through the solution. It says, create a new object called tongue_filtered, which contains cells with at least 25,000 total counts and genes that have more than 5 counts in at least half of the cells. So what we're trying to do is we have to construct a cell filter and a gene filter. And we have to use those two conditions to subset the tongue object. And we save it as tongue_filtered. So I'll leave that to the viewers. And we'll end the final minute. Keep an eye on, final minute. on how we can make some simple plots. So let us take our column data, convert it to a data frame, and call that object cell underscore info. So that's line 293. And so that's just the column data as a data frame. And if we use the head function, we can see the output looks good. Now, what we can do is, let me run this. So now that it's a data frame, we can use it to make plots using ggplot. So I'm just going to run lines 297 to 299 and see what the output is. So you can see that we have now made violin plots that show the distribution of counts for each of our 9 samples. That is correct. Yes. Sorry, sorry, it's a distribution not of counts, it's a distribution of total counts for the cells corresponding to each of the 9 batches. Right, that's the correct thing. So the way to do it is you use data equals cell info, so that's the name of the data frame. And then within AES, you specify on the x-axis your batches, so that's just batch. And Y, you specify, in this case, we want to look at the distribution of total counts. So you just do Y equals total counts. And to make a violin plot, I don't think we've actually made one so far. It's easy enough. Geom underscore violin, we give it this brown color, plus theme underscore black and white to give it a nice background. And then we do some tidying up. So from the book, it says, what if we wanted to visualize the distribution of expression of a particular gene in each batch? This now gets a little more complicated because the gene expression information is stored in the counts assay of our single cell experiment object, whereas the batch information is in the column data. This already is getting really confusing. To bring both of these pieces of information together would require us to do a fair amount of data manipulation to put it all together into a single data frame. Thankfully, we don't have to do that. This is where the SCATA package, so that's a package we installed, one of the many packages we installed today. And so this is where this package is helpful because it provides us with this function called ggcells that lets us specify all these pieces of information for our plot. So to see how ggcells works, let's go to line 307. Let's first just reproduce this plot, but instead of using ggplot, we'll use ggcells. So you can see all you have to do is replace ggplot with ggcells. And if we run this, we get an identical output. Great. I put question mark ggcells if you want to learn more about this function. One thing to know about ggcells is, so I mentioned this, as you've seen, the single cell experiment object can contain multiple assays. By default, if you don't specify the assay in the code, ggcells will use the assay log counts, not counts. Okay, so now we want to, the question was to plot violin plots for just one gene across the batches. And to do that, you can use ggcells, name of the single cell object. Oh, that's another thing that's different for ggcells versus ggplot. Instead of giving it the name of a data frame, you give it the name of the single cell object. And then we want x-axis to be batches, and y is the name of the gene. And the rest stays the same. If we run all of that, we now see the distribution of the gene counts across for the cells in each batch. Yeah, so this is so I would recommend that you go back to this and see what they're asking you to do. And just for the fun of it, try to do that without using ggcells. I think that would be a useful exercise. You'd have to do quite a bit of data wrangling. But if you want to avoid that, you can use ggcells like this. So that actually covers pretty much everything from this chapter. Next session, we'll dedicate it to the next chapter, which is on normalization, quality control, filtering. And then the session after that, we will dedicate that to differential expression analysis and clustering. So thank you for your attention. And please, if you have any questions, either comment on YouTube or email me, email any of us. Thank you for watching.